Transcription is when a strand of DNA is read to form an mRNA strand. It is the second step of the central dogma and consists of three phases: initiation, elongation, and termination. It's as easy as 1, 2, and 3.
1, 2, 5. Lisa, 3.
Transcription differs in prokaryotes versus eukaryotes. However, the beginning process is the same. The promoter signifies the beginning of the gene which is going to be copied, and it contains the prigno box known by the sequence TAT-AAT, a 10 nucleotide sequence upstream of the promoter, and the 35 sequence known by the code TTGACA, found 35 nucleotides in front of the gene that is transcribed. When these sequences are present in prokaryotes, RNA polymerase will bind to the DNA guided by the sigma subunit. When these sequences are present in eukaryotes, enhancers and silencers must prepare the promoter region for the arrival of the king, RNA polymerase. These can range in size anywhere from 50 to 1,500 base pairs, and they can be as far away as 1 million base pairs away from the site of transcription. Additionally, they are cis-acting, which mean they are in the vicinity of the structural portion of the gene. Then the king arrives and sits on the enhancers and silencers to begin reading the DNA and is again guided by the sigma subunit. Elongation begins with the sigma subunit disassociating when RNA polymerase opens up the transcription bubble by breaking the hydrogen bonds and building in the 5' to 3' direction in accordance with the synthesizing mRNA strand, attaching to the free 3' hydroxyl group with the nucleoside triphosphate, NTPs, acting as a substrates for the enzyme. Termination comes in two flavors, row-independent and row-dependent. For row-independent termination, the final sequence is identified through specific consensus sequences. These identical sequences fold over and bind to each other, creating a stem and loop at the end of the newly coded mRNA strand. The stem and loop stabilize the mRNA strand and kick off the RNA polymerase. For row-dependent termination, the protein in row binds onto the end of the gene and acts as a stem and loop to stabilize the mRNA from falling apart. Once the protein is attached, the RNA polymerase closes the transition bubble and detaches from the DNA, leaving a completed mRNA strand and a DNA double helix.
I hate goodbyes.